Note, welcome back. Our next speaker for the afternoon is Arvind Kumar from the University of Freiburg, and he will tell us about modulation of correlations in spiking neural networks.
Thank you very much. Thank you, Srini. Thank you, Sitabra, for inviting me in this nice festival season. This gives me a chance to come to India around Diwali time. That will be like after 10 years, so thank you so much. I'll be talking about correlations. Perhaps the idea was initially that I talk about basal ganglia and something, but I think that's a bit old stuff. So I thought I'll try to, tell something new, which is still in processing mode, meaning in review process and so on. And also link that to basal ganglia. So you can think of it as a long introduction to the basal ganglia function, if you wish. Before I do anything, I should thank people who worked with me on this. So a grad student, another grad student, a master's student visiting us, and Manny just finished her PhD, and my mentors, Ad and Stefan. Jens was a postdoc for some time with us. So what do? I like to study networks, and to me, if you look at brain, there are networks everywhere. You go from the networks that Janet was describing, biochemical cascades. You go to the functional connectivity in different behavioral tasks. That's also a network. So when I look at it, I see networks. And I just don't see networks. I actually see networks of networks. And just to give you an idea of that, if you look into a little piece of a cortical column, it may turn out to be a network. But if you carefully look at it, you see that different neurons, they are organized into layers. And you have 4 well-defined or 5 well-defined layers. And these layers are communicating with each other. If you go to columns, you say that this is one network. If you go to columns, then you will find out that columns are talking to each other. You can define the same thing in terms of processing of different sensory modalities or cognitive tasks or motor tasks. You can define hierarchies from retina all the way to the highest centers of visual processing. And there are then more well-defined structures in brain which are subcortical or at least not neocortex, like hippocampus is very well defined into different networks which are highly specialized, but still it's a network of networks. Then basal ganglia is a very nice example of yet another kind of network, and so is amygdala, cerebellum, and so on. So to me, a motif that stands out about brain is that no matter at what scale you look at it, you will find networks of network interacting with each other. And if you say that, then you can somehow make it an abstract notion of one network of excitatory inhibitory neurons connected in some way. You know the topology of it, and it is connected to yet another one, and so on and so forth. And if you do this kind of abstraction for the brain, you can forget about low-level detail, high-level details. You say that I develop methods, I ask questions at this level, and hopefully I can translate them back on the lower level, on the higher level. But either way, at this level of networks, I have learned something. And when I say that I have learned something, I want to ask 2 questions in this talk. One is about transmission of spiking activity, and another is how incoming activity interacts with the ongoing activity. So what do I mean by that? So if you have this organization, you have spiking activity in this network, this is going to go to the next network. And in between is this projection structure. And I'm going to argue that this projection structure is not just a one-on-one transfer. This itself can do some computing. So you need to know what kind of spikes can go from here to here, and do they change on the way, and if they change, in what way. And if you do not want them to change, then what you should do. Other thing is that once this activity arrives here, what's going to happen? It could change the firing rates, it could create oscillations, it could create everything else possible. But this question has to be asked not just on the side of input, but also on the side of the receiving network. That means ask a question, what's going on in this network where the activity is arriving, whether this network is tuned enough to receive it or not. So these are the 2 questions I'm going to address. And when I say interactions or, of ongoing activity with incoming activity, I will not talk about firing rates because I think we understand fairly well what happens to averages. What I will talk more about is the second movement of the ensemble activity, that is the correlations. So when you want to transmit some activity, I'm really sorry, I was very loud. So when you want to transmit activity, what you're interested in? If you look at a raster of spikes from some number of neurons, here 10, you can, I mean, this is a composite. This is, you could say that this is a response to something, and here you can already see that there was some modulation, and then after that there's another change. So what people often do is that they try to cut it down into different aspects, and they say that maybe this is one type of activity, synchronous activity with some precision, but there could be very, very precise activity patterns, and there could be completely asynchronous patterns where only the count matters. So these are the two sides I'm going to take for next 10 minutes or so that ask a question about what happens to this kind of activity and what happens to this kind of activity when I try to propagate it from one network to another network. So what are we looking for? So let's say that I have encoded some information in firing rates. By definition, firing rates means that neurons are spiking away in a stochastic manner, maybe in a Poisson manner. So you need more of them to average and get a good estimate of it. That means you do not want correlations between them. You don't want that two neurons in this raster, they have same spiking activity, because that doesn't help you in the averaging process. So rate propagation is a scheme where we think of it that if information is encoded in the form of rate modulations, then as it goes through different layers, it maintains its rateness, meaning it doesn't develop correlations. And if you have different rates for different stimuli, you maintain a unique relationship between them. But what happens in these networks is that you can also have a situation where this rate signal, as it goes through, it starts to become synchronous. Some people will call it that it's a nice rate to time coding, but because we started out saying that I encoded my information in rates and I want to recover it in the form of rates, then I would say this is a mode which perhaps you do not want. There is yet another way of thinking about activity propagation, as I said earlier, that maybe we are interested in not in the count of the spikes, but in the timing of the spikes. So if I have encoded information in relative timing of the spikes, can I preserve it, or does something happen to it? Or I could say that I just encode activity in this little synchronous volley of spikes. And as it goes through, whether it propagates or it diffuses out in the background. So one would be a failed propagation for synchrony. This would be a failed propagation for rate-like signals. A good way to think of synchrony propagation is in the form of this two-dimensional graph, just to give you an idea of it. Here you describe the time window in which you are counting your spikes. And on this axis, you count how many spikes do you have. And then you iterate it from one group to another. So you start out here, then you are here, and so on. So these kind of activity patterns, they just go through and end into a stable fixed point, or there is an attractor here. These points who do not propagate, they end up on the other side and diffuse away. So these are the two things that we can look for when we consider the simplistic picture for neural coding, in the form of rates, in the form of synchrony. So these two modes, obviously when you do experiments, you find them all the time there. Depending on who you are, how you analyze the data, how many neurons do you have, you can always take either side. So we were interested in finding out whether there are modes where you can have both rates and timing, both can exist together. And it turns out that, in fact, there's a very simple reason why things could go either direction, to the rates, to the timing. And that reason was the structure of connectivity. In these networks, what we do is that we assume just a layer of neurons which are unconnected, but they make projections from projection to the next group and so on. So what we find here is that depending on the projection density, so how many projections do you have, and what is the strength of the projections. These are two very crucial parameters here which decides whether the activity is going to propagate or not. So on x-axis here you have connection probability, on y-axis you have connection weight. And obviously these two are related things because you can compensate for one by the other one. So you would expect something along this line, a balance, that both are same. But still, what you find is that along these diagonals, you find that there are different regions where you can have either rate propagation or synchrony propagation. If you have a very weak connectivity, each individual synapse is weak, but many, many connections, meaning you are sharing a lot of input, so you are going to develop synchrony. So this would be a region where you will have synchrony transfer. If you have very few synapses, but each one is very strong, then you are going to maintain rates because there is no sharing of inputs. There is no reason that there should be correlations. So on this corner, you will find rate propagation. On this corner, you will find synchrony propagation. And in between, there's a nice region where you can talk about both rate and synchrony. And this was nice because it brings in the network structure, or in this particular case, it brings in the structure of projections into question, that this itself is computing something for you by transforming the activity. This is a very provoking statement, and we can discuss it afterwards, how much I can convince you on this. And later on, I'll show you more examples and more evidence in favor of this sentence. Then obviously what I did there is that I took very simple case. There were only feed-forward connections, only excitatory connections, and you can ask what happens if you have inhibition in this network. So same setting. So you have one group of neurons connecting to the next, and then next, and so on. But in parallel, they send out information or spikes to the inhibitory neuron. So you have a traveling excitation and inhibition together. What you can also do is that you can play around with the strength of the inhibition, and you can play around with the timing of the inhibition. So relative timing and relative strength of inhibition is important now. And what we find there is that if you represent a signal like this, that it has sharp transients, and then there is a sustained part. So this would somehow, qualify for a synchrony, and this will qualify for rates. It turns out, if you have strong inhibition, so just the weight is tuned, you can get rid of this component. What you will only propagate is the sharp onset transients. To get rid of these transients, what you need to do is you need to adjust the timing of it. So relative timing was important to get rid of synchrony, and the strength of inhibition was necessary to get rid of the rate component. But this means that we have now two parameters, and I can independently tune them. And if I do that, then I can create a situation where by playing around the relative timing and the gain, I can get either propagation of the synchrony, other way around, here, or I can get propagation of rates. And if I intersect them, I can create a situation where I can live with both of them. What was nice in this picture is that earlier with feed-forward excitation, we only talk about feed-forward networks as high-pass filters, that they only develop synchrony and that's it. And here you can play around by inhibition, change the gain of the filters, if you like, that kind of terminology. Next question was that, as I said earlier, that not only it is important what happens, not only it is important to know what you are sending, but you want to know who is receiving it and in what state that network is. To do that, we created a recurrent network where we can tune different states. So we can create a background activity which is very, very asynchronous, low firing rate. Everybody is uncorrelated with each other. Or we can create a situation where a lot of neurons are firing and there is a lot of dependence on each other, that is, neurons are correlated. And now you can look for, again, the propagation of synchronous volleys, how it depends on this background activity state. So I show you 6 examples which are taken from the 6 edges of this picture. 2 are in the middle and 4 are at the corners. What you find is that when everything is low firing rate, 2 hertz or less, very low synchrony, at that time, no matter what your signal is, you still get that behavior that you saw for the feed-forward network, that very weak inputs, they go and converge into the fixed point. As you change this network, as it becomes more synchronous, high firing rate and high synchrony, at that time this propagation doesn't happen. Meaning that when everybody is spiking together already and you want to enforce coincident firing on that, you can't really distinguish between that. Meaning this network is not ready to work with synchronous patterns because it already is synchronous. And to really impose information on this network, you need to do something else. Right? So what we concluded from here is that asynchronous activity, low firing rates, that's good. And synchronous bursts or synchronous oscillations perhaps are not so good. You can ask the same question. Maybe that you have synchrony, and this synchrony is more useful for rate signals, that you can propagate firing rates where there are no correlations. And it turns out that even rate signals, they propagate only for this asynchronous background, and synchronous activity destroys even the rate signal. So you do not want a network which has very high synchrony in that. What is good is like a nice calm surface of the, like if think of the analogy of water surface, you don't want big waves. Big waves are bad. If it's a nice smooth surface, you can do whatever. You can even see the responses to throwing in small pebbles in that. But this was really like a very abstract notion of what kind of inputs that could come in. In general, what you saw was that the signal was actually comprising of both synchronous activity as well as asynchronous activity. So you're more looking for something like this. That you have a spike pattern coming in, which has both information in its count. It has information in terms of the dependencies of the spikes with each other. And our question was how this network is going to respond to this kind of input. We were hoping that we have learned a lot with these very simple signals and we can sum them up and maybe that helps. But it turns out, at very first step, you stumble. The question was, how are you going to input these correlations? Who receives them? It could be that all these spikes, they go to one of the neurons and then you are done. Or it could be that each one gets its own unique spike train. That means you have lost the information that was there in terms of the relativity or in terms of correlations. So one could imagine this kind of scenario that a bunch of spike trains, they end up into these blue neurons. Other bunch ends up in the red neurons. But if this happens, then you need to separately define the correlation and the rates for these boxes. Meaning you need to define what is the correlation within these 3 spike trains that are going into the blue neurons. And you need to define it separately for the red neurons. Now, by construction here, I didn't know this, that where these spike trains are going. As an experimentalist, I only recorded them. Meaning, I also need to define correlation between these 2 groups. Right? So that means to understand correlations, you need to go one step further. It's not sufficient to say, I have 10 hertz firing rate and average pairwise correlation is 0.01. That's insufficient. You need to say that who is receiving them and in what way. So a general picture that emerges from this thing is that if you want to understand how a network is going to respond to correlated inputs, you can describe it in the form of this very simplified picture. There are 2 neurons. You need 2 neurons to describe correlations. If it was only a rate story, then we would have done with just 1 neuron. And then you have local connections of these 2 neurons where there is some correlation that are going into 1 neuron, which is in these cyan arrows. So this we call as the within correlation because this is private to a neuron, and you can think of it as if this is the correlation among the spike trains presynaptic to a neuron. And then these 2 guys, they can be correlated themselves. So this is a between correlation. So correlation between the presynaptic pools of 2 neurons. Okay, so within and between. And same thing now occurs for the stimulus. So now think of it as a visual system. So this would be the recurrent connectivity in layer 2, 3, and this is the input coming from layer 4, let's say. Or other way around, this is layer 4, this part, and this is thalamus. So from the input side, you get, again, within correlations and between correlations. A more simple way to think about it is that you have convergent projections that create within correlations, and you have divergent connections which create the between correlation. And it turns out this distinction is very, very important to understand what these correlations will do. Early on, already in 2003, we knew that correlations, they can affect the firing rate. So here there are 2 different models, but in both models, depending on how you create spike patterns, you can get increase in firing rates. That's no big deal. We understand it, why it happens. There was another interesting point that came out was that the transfer function of 2 neurons now obviously depends on the input correlation, that more input you give in, more output correlations you will get. But what they also observed is that this output correlation depends on firing rates. And firing rates in terms of 2 variables, because they were working with noise, they could define the average of the noise and the fluctuation size. And we know that both are going to change the firing rates. So by increasing this mean and the variance of the input, you could change the transfer of the correlations. Obviously, you will not get more correlations than you put in, but still, efficacy of that transfer can be changed by changing the fluctuation size. We do the same thing here, and we ask a question now. Let's say that there is this one group of neurons which feed into two neurons, and there is some within correlation and some between correlation. What this will do? We expect that within correlation, because this is only private to a neuron, this is not going to affect the output correlation. It will only change the firing rates. Between correlation is the one, this is going to translate into the output correlations. So you have between correlation, output correlation, and what you see is the same thing, that you get some output correlation, but you can modulate it just by changing correlations within these groups. So not difficult to understand, because these within correlations, they are changing the fluctuation size in the neurons. Bigger the fluctuations, larger is the probability of spike, and then you will have coincident spikes. So the idea was then that effect of this B correlation actually depends on the W correlations. You could have a very high degree of divergence, but if each one is very weakly correlated, it's not going to do anything to that. So to understand it more, those who follow it is easy, otherwise you don't have to pay so much attention on it. The idea is that you can do some mathematics for this, because you have created this very reduced description. You can think of two neurons getting some shared input and some non-shared input. So each neuron gets a mean current, and then on top of that it has the fluctuations which are shared. So there are independent fluctuations which are private to this neuron, and there is a shared component. And same thing you can do for the other side as well. And by changing these two, now you have the description of the statistics of the input currents. That translates to output, of course. So by calculating correlation between these input currents, you can say something about the output currents. That's the basic general framework. Or in more pictorial way, that same equation is here, that you have a neuron which sums up input coming from independent sources, but it also receives something from which is shared between the two guys, and same thing from the other side. So this would be, let's say, the recurrent side, and this is the input side. And in the end, what you are going to do is that you are just summing up these white noises, creating your distribution, and your problem is to find out the correlation between them. And we do that because now it's relatively very simple. What turns out is this little, I don't want to go on this, it's not so much anyway. The output correlation depends on B1 and B2. B1, remember, is the correlation. B1 would be the correlation here, and B2 would be the correlation here. So it depends on the input correlation. It depends on the existing correlation in the network. Also, there is a little factor S which decides the relative fraction of these two. So if you change these, then you can get an interesting picture in terms of correlation modulation. So let's say this is the original case, that this is the ongoing activity. It has some correlation which increases, and obviously there is no S, meaning there is no stimulus coming in, so nothing changes on the y-axis. But now you start to bring in external input. As you bring that in, you see that here you are not doing much. You are changing slightly the color here, but what is interesting is that at this point you had high correlation, and now you have reduced those correlations, right? If I take a subtraction of this and this, I find out that indeed this is what happened, that in this part where I had strong input local correlations, I can kill them by having some external correlated input, right? So the upshot is that here is a way by which you can bring in correlated input from external sources, and if the network is correlated, you are going to decorrelate it. On the other hand, if the network is already uncorrelated, the baseline is uncorrelated, then these correlations have meaning, right? So the modulation or the output correlation in the network, they depend on the input activity, of course. Also, they depend on the ongoing activity. And here what I did is that there is this flag here which says that variance is matched, that I was careful that I do not change anything of the mean or the total variance of the inputs. Let's say that I relax that, because really in real situation a stimulus doesn't care that it wants to keep the firing rates in the output constant as it were before. It changes the firing rates, right? So let's allow this guy to change the output firing rate. So now you will have two variables, firing rates and correlations. I will not show anything about firing rates. If you do that, then you have a slightly different transfer function. Output depends on B1 and B2, but it also depends on the local fluctuations. So the convergence pathway, which was creating the within correlation, that becomes very, very crucial because it figures here. And that's then the correlation that comes out. And this is your comparison line. That's how the correlations are without any external input. And what you will see is what's happening in these traces. So this is for 1B1 and B2 and so on and so forth. If I do the subtraction from that baseline, I get again the similar picture. So B1, B2, they matter. W1, W2, they matter. And in the end, what you have is this nice landscape where by changing the fraction of the local fluctuations now. So when I'm changing gamma, I'm changing the relative size of the fractions. I'm not changing anything in the B1 or B2 that way. Only thing that changes is gamma. And if you follow one of the pixels, you will see how it changes the effect of input correlations. So the upshot of this story is that to understand how correlations affect the ongoing activity, you need to know projection structures, you need to know what is the ongoing state of the network, and you need to know what you are feeding in. So without these three, your description is going to be incomplete no matter how hard you try. And that's exactly what I have just said. So input correlation, projection structure, and the ongoing activity. So now, with this long introduction, I can start to ask something more, some interesting questions. And in this case, I did it for basal ganglia. And the reason is that it's complex network, but it's also simple. The idea is this. How much time do I have? 15. Okay, so the idea is this, that it's amazingly complex network. It does amazing function for you. Ask the people who have Parkinson's disease, they will tell you how crucial it is if you didn't follow talks of Stan or Janet. It's very important. But on the surface level, it looks very simple that you have striatum, which is all inhibitory. There's no excitatory input in this. You have globus pallidus, all inhibitory, excitatory. So in that sense, you can do a little bit of mean field version in terms of calculating something for this network. And obviously, this network is very, very simple. It's all inhibition. We know a lot about inhibitory networks. So that's why this was the choice. But what I will show is that these correlations, the way I have defined them, have very interesting effects within striatum, creating something related to Parkinson's disease, and also maybe in terms of the decision making. So let's start. So we did this game of B's and W's, that is between and within correlation. At that time, we didn't think so much about it. So at that time, we used rho and C for these values. Very innovative. But the idea was again this, that you have groups of neurons in cortex which project onto striatal neurons. And the idea is now that you have in each group there is some correlation, and also there is correlation between the groups. And what you are interested in is that how this thing changes the response function in striatum. So what we did is that you have ongoing activity in the striatum, and then you give it a step. So it's a Poisson input, so it changes the firing rates instantaneously for 100 milliseconds and see what is the response. And what we are looking for is the ratio of the response with the background activity. Because it's an inhibitory network, we say that the most rudimentary thing it can do is contrast enhancement of the signal, because there's a lot of recurrent inhibition. And that we define in terms of signal to noise ratio. Signal to noise ratio says how much is the signal in the stimulated neurons and how far it is from the background activity. And this is then the color. So obviously the correlations here within the group, they influence your signal to noise ratio. But what was very interesting is that if there are correlation between these two groups, then the signal to noise ratio decays. What does it mean? It means that when striatal neurons, they start to share their projections from the cortex, then the contrast enhancement doesn't work that effectively. And lo and behold, it turns out that this is how the connectivity is structured from cortex to striatum. This rho is indeed very, very small. It's not absolutely zero, but it's not very high as well, which you would expect by definition because of the numbers that are in the two populations. So this was very nice that the distinction makes sense, and you can comment about even the significance of anatomy. Next, we ask a question about role of basal ganglia in oscillations. And that time we were working on models of Parkinson's disease-related oscillations. As Dieter said, everybody has their own model. We have our own. And in this case, this is my favorite one for sure. Our idea is that you have these oscillations here because, I mean, obviously this circuit oscillates, but these oscillations are unleashed, so to say, by increasing the striatal firing rates. That's our mechanism, that it should work this way. So we did show that it indeed works, but the question was that can we do something more in terms of correlations for this? So what we did is that created a similar picture, but this time not for striatum, but from striatum to globus pallidus. And once again, you are talking about correlations. No change in the firing rate, just change in how spikes are coordinated. And these are two examples. So here there is a very, very small correlation, if at all. And what you see is almost flat firing rate, no trace of any oscillatory activity. If you introduce correlations, it's not so well visible, but if you sum up over the population, you start to see these nice oscillatory activity states. So just by coordinating spikes in striatum, you could create these oscillations. And it turns out, in this particular case, it doesn't really matter whether it's between or within. As long as any one correlation increases, there is a general tendency to increase towards oscillations. We know that increased firing rate is demonstrated in striatum during Parkinson's disease. I don't know if there is literature on correlation state, but this would be then a prediction from our side for Parkinson's disease model that it may be that the onset is not visible in firing rates, it's just an overall increase in correlation state of the striatum. The next question was about the go and no-go pathways. The basic idea is this. It's, I think, one of the simplest possible description you can think of for such a complex circuit. There are these neurons, D1, they project to globus pallidus exclusively. There are D2, which project exclusively to GPE and then to GPI. So people have defined these so-called direct pathway, indirect pathway, and hyperdirect pathway. And this pathway is overall positive. Indirect pathway is overall negative. And hyperdirect pathway is also overall negative. And people say that these two things are responsible now for all your action selection and everything else. One can work out how it will be and so on. Because this is, I think, the really primitive description of it. Hopefully you can build more complex things out of that. Now, what it was that you want some activation in direct pathway for go, and you want some activation in indirect pathway for no-go. Things were easy until 2008, and in 2008 they really demonstrated that there are D1 neurons and D2 neurons, they are separate. Not only that they are separate, in 2010 they showed us that the connectivity is very different. D1, they have less recurrence, D2 have more recurrence, but also they have strong connections back to the from D2 to D1 is very high connectivity. The question is that how are you going to deal with this if there is so much of connectivity from D2 to D1 as opposed to D1 to D2. So it's not really like one activates, another deactivates. Like Stan showed in the morning that there were inhibitory connections between two networks. If one activates, other shuts down. Here it's not going to happen in that simple way. So we asked this question first for the cortical firing rate. So as a function of cortical firing rate, what's the response in D1 or D2? So that's on this axis. Red ones are D2, blue ones are D1. And these different shades, they refer to different amount of dopamine. It's a very rudimentary model of dopamine, I have to admit that. And now what we are looking for is that where are the transition points where D1 dominates D2 or vice versa. So for instance, this is the point where D2 activity becomes higher than D1. If you change the dopamine level, you can actually change this point where the transition happens. So a very simple idea would be that if you have lack of dopamine in your system, then you need really strong cortical input to get the D1 guys activated. right? Only here you are starting to cross them over. Oh, sorry, here. So this is one possibility. Second possibility was, of course, I mean, correlation is my favorite word in neuroscience, that maybe there is something more you can do, that you don't need to change the cortical activity. You can just do with the correlation. So here, what we have is the difference between the two firing rates as color. So lambda d1 minus lambda d2. So positive means d1 is higher, and negative means d2 is higher, and negative here also means that you are jammed. Basically, this is the no-go pathway. So as you change the within correlation now, so remember these are the correlations which are private to neurons. You can switch for the same amount of firing rate. I think we held this picture for 30 hertz, so somewhere here. So if this was the transition point where D1, D2 started to alternate their roles, then you can change that. And all you need to do is that change the local correlation coming into the neurons, which is here. So if you go from point 1 to point 3, you have already flipped the balance. Once again, B, they have a very interesting role. We don't quite understand it, but still, the general picture is that, first of all, they ruin this differentiation. Second thing is that there is some other interesting nonlinearity here, which is that here this difference increases, and on this side it decreases. This is something we don't quite understand. But nevertheless, the point is that you have this D1, D2, and you can create a competition between them just based on the correlated input private to neurons. Although I hate it, but I mean, I'm a person who talks about networks all the time. And here I think I actually bring in the single neuron behavior, because I'm talking about correlations coming into a single neuron, and then single neuron firing rates can dominate the effect. So I think this is a nice way to go backwards, and this is, I think, a good way to think about brain, that you have links to lower level and links to higher levels. So all this story, but you can, I'm sure you would have asked, if I asked you to ask me questions, then you would have asked, well, you have made this nice story about W's and B's. What's the evidence that they are there? How are you going to differentiate between them if they are there? So what we did is that we created network simulations and try to find out whether we do get some difference in B's and W's and so on. So these are different connectivity matrices. So we have random connectivity, mildly clustered, or very, very clustered local connections. And these are two samples of activity. This is a random network, and this is a clustered network. Both have same firing rate, but in one you get this kind of stripy behavior. So each row is a neuron, and each dot is a spike, and that's a raster. So to find out W's and B's, what we did is that we have this arrangement, and we look at all the connections of a red neuron or a blue neuron. So that would be within correlation, red-red or blue-blue, so in this schematic. Or we are talking about between correlation, meaning a neuron which is connected to red and a neuron which is connected to blue. And clearly difference is right in front of you, that B correlations do not change with clusters. Different colors here means clustering. So if you become more and more clustered, what happens is that you start from very small values of W to very large values of W. So if I put them together, this is a random network where within correlation and between correlations are comparable. There's no difference. So if you believe cortex is random, brain connectivity is random, then whatever I have said, you can forget it. But if there are clusters, then depending on how clustered the networks are, you can have very small value of B, but very high value of W. So B's and W's, they don't span the full space, but nevertheless, there is a difference between the two types of correlations. And we know now that W correlations are very crucial in improving the transfer of correlations in terms of deciding how much modulations you are going to get. And this has very strong implications now in terms of how we are interpreting activity. I think I have mixed up my slides. Yeah. People talk about correlations. First is that they randomly pick neurons and correlate them. Other way to talk about correlations is in terms of what is called signal correlation and noise correlations. Idea is that there could be neurons which just, you present the same signal again and again, and they give correlated responses. That's called noise response, noise correlation. And if two neurons, they have, let's say, this kind of tuning curves, these two neurons, if you present different stimulus, they will have correlated firing rates because of their tuning curve preferences. And these are called signal correlations. Turns out, this is just one way to think about correlations. What I tried to argue is that this perhaps doesn't even matter because you are talking about over trials, different presentations of same stimulus. This is about decoding. But if you are interested in understanding correlations' impact on the network, then you need to talk about W's and B's. And what is nice is that, okay, difficult part is that how are you going to know ever who is connected to whom? This detailed connectivity we will never know, or not to the extent that we need here. But what is nice is that because I said W correlations are private to a neuron, you can read the signature of W correlations in the membrane potential fluctuations. And that's what we show here, that depending on what is the value of W correlations, you can get some mapping between the standard deviation of the single neurons. So by looking at the single neuron membrane potential statistics, you can talk about what extent W is existing, how much is B by comparing two neurons together intracellularly. And I think I'm running over time. And just to summarize whatever, okay, perfect. So just to summarize what I said, what I was interested in is that how networks communicate. And I argued that what we need to know for that is three things. What is the input, what is the projection structure, and what is the ongoing activity. And in that process, what I ended up doing is that I ended up defining correlations in terms of W's and B's. W meaning convergent, if you wish, in a simple way, and B meaning divergent. So convergent, divergent pathways, they are everywhere in brain, meaning you can always define W's and B's. And as soon as you define them, you open up the range of correlations axis. Earlier you had one axis, now we have actually four axes. It's a four-dimensional space, and you can navigate through it by creating interesting modulation paradigms for correlations. And that itself can become information processing. But beyond this abstract notion of correlation, they're used for function and so on. I gave you three examples where they do drastically change the behavior of a network, like for instance, changing the preference of D1, D2 neurons, or creating oscillations even. So obviously now the ball is in the court of the experimentalists to really measure these things. Already the data that is coming out about connectivity, that kind of supports that it's at least not random. There are interesting structures. So maybe we have a chance. Thank you very much.
In this two neuronal system model that you have, when you're looking for in between and within correlations, so if you were to think of visual input, for example, would you hazard a guess as to what the data processing or information processing system really is? Is it in the correlations? Is it in the spacings between the spiking patterns? Where is it? What is it?
So if I look at the natural scenes, there, what I know for sure is that there are spatial correlations, there are temporal correlations in the scene. And then that means already at the input level I have correlations. And then I have projections from similar neurons in LGN to V1. So that means this convergence is going to create a situation where I will pass on the stimulus correlations into V1. There is also divergence, so I create B as well. So I think if one tries to map the structure of thalamocortical projections, we could redefine the stimuli in terms of B's and W's. And I'm convinced that every natural scene would end up giving you some correlation picture. So extending this argument, I would say that visual information is in both, in terms of tuning of the neurons, which neurons are activated, but that specific feature-related information is in the coordinated spiking. No, I'm not root for them, and I'm actually on the fast time scale synchrony. We are not talking about rate-based correlations in this. And I'm not even sure whether I can talk about that right now. But yes, in this picture, whatever I talked was about fast timescale correlations up to 10 milliseconds or so.
Well, 10 milliseconds going to 1 millisecond.
Yes, of course. But let's put it this way then. I'm talking about correlations which are relevant for neurons given the knowledge of their time constants. Whatever could be integrated in one time constant, let's put that as my time scale. There are these slow variations, and in fact, in these pictures as well, I'm very careful when I'm saying I don't want to talk about it, because what happens is that although we change the correlation peak, we actually, I mean, if you look at the full cross correlogram, right now what I talked about was the peak, and I could go further out and I can sum up over long distances, and their picture becomes difficult because correlation peak could also go down. So I don't know how to calculate them even. So this is why I don't want to get out of the first dominant peak of the cross correlogram.
On that side, any particular from each village, it's a slightly different track than your synchronous.
Well, I think polychrony is, I mean, neurons have to spike and somehow they have to integrate inputs. So yes, I mean, polychrony has to exist. I think it's, I mean, just because you have different delays, you have to have polychronous events, otherwise neurons will not spike. So I think it's a very ubiquitous thing in the system. Now, whether it can be exploited for some coding purposes, I don't want to say, I don't know actually what I will do with that.
Just a short question, Narendra. How would you extend this analysis of basal ganglia to develop a model of a task that basal ganglia is involved in? So this is a very interesting theoretical result.
So I think we are slowly taking steps in that direction, but I have to say that our view on this is that, start with what this network is capable of doing. Right? So I have some hardware in terms of D1, D2 connectivity, GPE, GPI patterns, or SDN having a control. So right now we are in a situation where we are just exploring the space, what we can do with it. And D1, D2 is just one way. I mean, you can also define other parameters by which you can modulate. So once we know this, I think it will become much easier to represent tasks once you know the complete dynamical picture of this system. So right now we haven't really talked about specific tasks.
You need to bring in learning and dopamine and all.
Yes, obviously. And that's why I was asking Janet about the dopamine effect on learning. And in these networks, we can't deal with that kind of complexity. Sorry. We need reductions. So.